Yeah, I think this is I think it's a tricky question. I'm not sure I have any kind of particularly novel insight to offer. I mean, obviously the big ones are still things like NK and CAR T cells, and I think we're still very much on kind of the upswing of seeing what those types of therapies can do, and I think probably the sector would largely recognize that. I think there's some interesting kind of fusions going on in the space between kind of some more of the more kind of conventional biologics approaches and symbio approaches. So you have, for example, people engineering CAR T cells that the cells themselves are able to seek out cancer cells or whatever it might be. But then the cells are also engineered to do things like produce bispecific antibodies, which you've then got kind of a parallel approach where bispecific antibodies in themselves are a pretty well established and versatile kind of platform, and then you're marrying it up with CAR T, which is also showing to be quite promising in the space. And so I think we will continue to see a lot of these kind of innovative fusions of different approaches, and then you've got, as I say, you've got these kind of parallel approaches where you can potentially use two or three or however many different mechanisms concurrently to address some of the potential fallbacks of any one of them in isolation. I think probably one of the other big trends that is particularly relevant to us, based on kind of the questions that we get a lot, is that I think a big part of the sector is looking for alternatives to viral vectors, for various reasons. That they are obviously pretty successful themselves, but they're costly to produce, they're difficult to produce, there are immunogenicity issues with them, and a lot of safety concerns and kind of red tape and hurdles you have to go through. And so I think kind of based on the questions that we get, as I say, a lot of people are looking for different ways to deliver biological payloads, particularly nucleic acids, and in the case of viral vectors. And so I think we'll see a lot more activity kind of in our space, in the sort of novel drug delivery type space for these different modalities, or at least a lot of iteration on what's already gone before with viral vectors. And I think the other big one probably is that I think sort of the likes of viral vectors have already proven, I think, pretty conclusively that they can work. But now I think that a lot of the industry focus has to be on the support network to actually make these available and reproducible and safe and all that, the usual stuff that you have to address. So I think and as I say before, part of people looking for these alternative delivery mechanisms is about the cost and the difficulty of manufacture. So a lot of the activity I think that you'll see, and probably already are seeing, really is innovations and iterations on ways that you can get the scale up, the manufacture, the QC, and all of that good stuff for these types of vectors down. So I know there's a lot of companies that are doing things like designing and producing bioreactors for eukaryotic cell culture that scale better, for example. And I think synthetic biology has a lot to offer in this space. We're doing things like creating viral vectors that aren't quite as immunogenic, for example, that kind of thing. So a lot of it is, as I say, I think going to be in and around the support network to make the approaches that we already have and are beginning to kind of yield results, to make them just work as optimally as they can. Yeah, I think that's a really interesting question, and I've seen other people argue from both sides that there's both like too much money and not enough money, and it obviously can't be both. So I think with the the numbers that have come out in the last couple of years with sort of investment into the biotech space, I think it's difficult to say that there isn't enough money at this point. Like there's more money coming into biotech all the time. So I don't think it's that the sector is in any way kind of short of investment, but that's not to say that more isn't always welcome and advantageous. But from kind of our own experience, I think the sort of the age-old kind of valley of death, I think, is still kind of alive and well. The bit we really struggled with ourselves was the translation sort of at the very edge of academia, where an idea is deemed too translational to be kind of fundable from a research council perspective, and is kind of detached from the fundamental biology. But the ideas aren't quite mature enough for even very early stage investors to kind of jump into. And I don't think that gap has been totally bridged. I think it's probably better. You've got a lot of accelerators and things like that, which try to fill that space. But in large part, a lot of the accelerators don't offer anything like the type of amount of kind of prize money that like a company like us could really do anything meaningful with, for example. So I think there's plenty of money. I'm just not sure it's where it needs to be necessarily. And I think you need a lot more. There are a lot of ideas that are coming out of academia that never quite managed to attract that first bit of investment. And in some cases, that will be for the right reasons. The technology wasn't right, or there wasn't a market for it, or whatever it might be. But in a lot of other cases, it's just because, it was the same challenge I had, that you've come out of your PhD, you've got to go and find a job. If your innovation is attached to the research you were doing in your PhD, as it was with me, the minute you have to go off as the kind of innovator, if you have to go off and take a job because you've got bills to pay or whatever, chances are that idea probably dies, unless there's other people in the lab, behind you that are willing to kind of run with it. So giving people the space to develop those ideas sort of post PhD in very, very earliest stages is still tricky, I think. I mean, I spent a few months unemployed at the end of my PhD because I was waiting for a grant application to come in so that I could stay focused on this, because I knew that if I went off and took a different job, that would be it. The company just would cease to exist, because there was no one coming through in the lab that wanted to take the idea on. So I think, I don't know whether it's private sector money or whether it's public money that needs to bridge that gap. It's probably a bit of both, but I think that gap is still there. And yeah, I think the short answer from my perspective is that there is plenty. It just needs to be distributed in increasingly clever ways, probably. I think, to be honest, I would say I think the sector's priorities at the moment are broadly right. Probably the biggest one being, what I alluded to before with kind of the manufacturability and scalability. I think that's going to be absolutely key. There's no way the NHS, for example, is going to roll out therapies at £1 million a dose in a widespread way. So the costs really need to fall, or we need to find alternative ways of achieving the same thing that are cheaper. So, and obviously the pharma companies and the developers need to be able to make a profit on this. So the therapies are expensive, and the payers aren't going to meet that ticket price. So the only way that you can do it is that your production costs and your development costs have to be lower, so that you can sell it lower and you can still make the same, a decent or the same margin. So I think that absolutely has to change, and I'm sure that it will. I was talking in another interview I was doing the other day about a good example of this, even in just our own space in kind of SynBio, is something almost seemingly as trivial as like DNA synthesis. When I started my PhD, we looked at having one of our platform sequences synthesized. These are plasmids of about 30 kilobases. We were getting quotes at the time, so this would have been 2014, 2015, of, I think the cheapest one we got was like 18 grand to synthesize this plasmid. And even then, the supplier wouldn't guarantee that they could actually achieve it, and it would be an R&D project. Fast forward, 6 or 7 years or so, and we're now in it, just at the moment, having constructs synthesized for just a couple of thousand, where up to 30 kilobases in a plasmid is sequence guaranteed. It's turned around in a month, and so even just little things like, well, little in the grand scheme of things, but it's still quite an impressive achievement. Little things like that are obviously transformational to us, and so in the space of 30 years, I think that it'll be a totally different picture. How cheap you can get something like a viral vector or cell and gene therapy type approach, I have no idea at the moment. Obviously, you're never going to be probably at small molecule money, but certainly the costs, I'm sure, will tumble in the next 10, 15, 20 years maybe, as those innovations come through. And I think probably the other part to go with this would be that with advanced therapies in general, I think the regulatory landscape is going to be pretty different to your more conventional therapies. So I think having a quite progressive approach to this, to how we regulate these and what the sort of critical metrics for these types of things are going to be, will be really key. But I mean, fortunately, I think we've seen in the last 18 months that with like the mRNA vaccines coming through, that when the need is there, the regulators are very, very progressive, very sensible, and so again, I think the priorities are right, and it's just a case of kind of keeping those conversations bubbling along, really.